Okay.
All right. Any questions so far, folks? Okay, we have to ask ourselves the questions of how do these things, these specific transcription factors, interact with the basal complex. We said we can have direct binding to the basal complex, and that can stabilize or disrupt it, usually by working with one particular member of the basal complex. It either holds it in place or tries to push it off. So that's one kind of thing, the direct binding trick. Okay, so here we have, let's put promoter here. And here's your gene. Okay, here's our basal complex. Now we have an enhancer over here. We bind some kind of protein. One end of it binds to the enhancer sequence, and the other end binds to the basal complex. Now you have to be fairly close for that to happen, but it can do that. And that's going to be an example of direct binding. It may stabilize the basal complex, preventing it from falling apart, or it may actually disrupt it by pushing one of the components out of the way. But we can also do things indirectly. Now we said that some of these enhancers can work to and possibly as much as 3,000 base pairs away from the basal complex. Now 3,000 base pairs, that's a long physical distance. That's probably 100 nanometers, 200 nanometers. That's a pretty good distance. There are very few proteins that are so long that they can stretch that kind of distance. So that's where you're going to get the indirect interaction. Now one thing we've seen pictures of this in electron microscopes and stuff like that. On a short scale, a DNA molecule is very rigid. Picture a filled up garden hose. Okay, on a short scale, it's pretty rigid stuff. On a long scale, though, DNA is quite flexible. So here you go, the garden hose analogy is a good thing. You can have a nice filled up garden hose. If you try bending it over a little tiny space like this, it gives a lot of resistance. On the other hand, you can take that 100-foot garden hose and snake it around things and stuff like that. On a longer scale, it's much more flexible. Same thing with DNA. Okay, so we can have the DNA bending around. Here's an enhancer. We can have another transcription factor binding directly, even though this enhancer is located hundreds or even a couple thousand base pairs away. So we could do this. Those are examples of direct binding. But we don't have to bind base directly. We can do indirect binding through adapters. So let's kind of do a few things here. I'm going to stick, I'll put this back here. Put them back. We'll put an enhancer over here. Okay, different. Okay, here's our transcription factor. And here is an adapter protein. Now that adapter protein allows this effectively reach over and touch the basal complex and do something to it. It could be a set of adapter proteins. Here's another one. Here's transcription factor, and here's another adapter. Okay. So that's the example of indirect binding to the basal complex. Either way, it's going to affect elements of the basal complex in one way or another. Now, we take a little look at a couple of these types of things. It turns out that there are some widely used adapter proteins or complexes of adapter proteins that link numerous different specific transcription factors to the basal complex. So we can take a look at a few things. of them is, they sometimes call these collectively co-activators. In other words, they activate transcription in association with something else. One of the most common kind are what are called the TBP-associated protein, or TBP-associated factors. Here, I'll write some. Or TAS for short. Now, let's take a look at this. Here we're going to have our base. Here's our promoter. And here's our basal complex. Now what we're going to have here, we'll put the Tata box in. And there's our old friend, the Tata binding protein, the cornerstone, and here's the rest of the basal complex. The Tata binding protein, once again, that's the cornerstone, the foundation of the basal complex. It's the first thing to come in, and without the Tata binding protein, nothing else is going to assemble there. So needless to say, the TBP is a very important protein. Now it turns out, aside from binding stuff in the other components of the basal complex, the TBP binds several different types of proteins called the TBP associate factors, or TAFs. So what they do, and these proteins are large in molecular weight, they're usually a few 100,000, and they're fairly elongated, they're big molecules. And there's several different kinds. So they reach out and they're binding to the TBP. Now this entire complex of the TBP and the TAS. Now the TAS, by the way, are not directly part of the basal complex. They're associated with one of the critical proteins, but they're not really quite part of the basal complex. Now the thing with these TAFs, there's several different kinds that can bind to TBP. They're all big proteins and elongated proteins, so they can stretch out a pretty darn good distance. The complex of TBP and these TAFs, we call it TF, transcription factor 2 for polymerase 2, TF2D. So you see in the literature somebody talking about TF2D, it's referring to the Tata binding protein and its associated TAFs. Now this thing is a hoss. It's big, its molecular weight runs about 1 million or so. It's huge. It's really, really big. Now the cool thing about these TAFs, each one of them can bind multiple different specific transcription factors, depending on what happens to be around. So if I'm going to have to erase this a little bit. So let's take a couple enhancers, distant enhancers here. Here's a few of them. A little small one over here too. Okay, a specific transcription factor of one kind binds this TAF. Now we could have another one on this enhancer that binds to a different part of the same TAF. And another one that can bind over to here. So each TAF here can bind multiple different transcription factors. This is another reason why these are large proteins, because that allows them to have multiple binding sites for many different proteins. So many different specific transcription factors can bind to the same TAF, and that's going to have an effect. And look what these guys are connected to. Those TAFs are connected to the most important part of the basal complex, the Tata binding protein. If you push that sucker off of the Tata box, guess what's going to happen? The whole basal complex collapses like the U.S. economy did several years ago. It just goes down the toilet. Kiss transcription bye-bye when you do that. On the other hand, if that is a stabilizing interaction, holds it down real tight, the rest of the basal complex is more likely to assemble and hold together. So transcription shoots way, way up. That's one kind of thing. So our TBP associated factors, or TAFs, are important components that allow multiple different transcription factors to interact with the most important part of the basal complex, or our Tata binding protein. That's one thing. And then there's something else that we know a little bit less about this than we do about some of these others. It was first discovered in yeast, a gigantic complex of proteins, 20 or 30 different kinds, forming this complex with a molecular weight in the low millions. And people notice in the absence of this thing, it affected transcription in one way or another, usually by decreasing it, but not always. So they call this big complex first discovered in yeast the mediator complex. And when they looked in things like vertebrates and human cells and stuff, sure enough, we got this too. The mediator complex is another major thing, and ours are even bigger and more complex than ones in yeast. You're talking about, maybe 30 separate protein subunits. And apparently, it binds to the basal complex at one end. And affects things in different ways. And meanwhile, a whole slew of specific transcription factors can bind to it. Each one of them are going to somehow affect the mediator complex and thereby affect the basal complex. Now, so this is an important one in regulating transcription. We don't know huge amounts about it yet, partially because it's such a large, complex assembly of proteins. But in a sense, if we want an analogy here, all computers now have USB ports, right? And you can plug any of a number of devices into a USB port. Picture the USB port, a single USB port, being an enhancer. You can plug into that USB port a printer, a monitor, a game console, a digital camera, whatever, a flash drive, whatever. You can plug several different things into the same USB port. Just like you can put several, in many cases, several different transcription factors can bind to the same enhancer. Now, what you do at a computer, if you've got like 6 USB ports and you've got 10 things you want to hook into the computer, you go out and buy a new computer? Of course not. You go out and buy what they call a hub. It's a single connection into a single USB port, and then multiple USB connections, so you can run several different things through the same USB port. You can have a printer and a digital camera and an external hard drive and a monitor and stuff, all in the same USB hub. That's what the mediator complex, and to a lesser extent the TAFs are like. They're like USB hubs for different transcription factors. You plug in any of a number of specific transcription factors into our molecular USB hub, and then it goes into our USB port, the basal complex, and affects transcription. So one nice thing about this, both the TAFs and the mediator and all the other adapter proteins that might be involved. One nice thing about this, not only does it allow things like enhancers to affect transcription a significant distance away from the promoter, but it also allows a whole bunch of different things to affect the basal complex, far more than if you're just dealing with direct binding. Because, big as the basal complex is, there's a certain limit to how many things can bind to it at once. But if you have these huge complexes here that can bind multiple different proteins at any given point, that's going to have all kinds of effects. So in other words, these kinds of things not only allow transcription regulation to occur a good distance away from the promoter, but it can also allow a huge diversity of different transcription factors to ultimately affect transcription by dealing with the basal complex. Okay, that is some general principles of specific transcription factors.